So let's go ahead and get started. Overheard a couple of questions here at the front about what's going to be on the midterm. Everything since the last midterm. That's the second half of Chapter 5, so everything which is not replication, all the repair stuff and recombination, as well as Chapter 6, so transcription and translation, which we'll maybe get started with today, but definitely finish up for the rest of this week. Any other questions about the next midterm? It feels like we just finished the last one. I know. I will be trying to upload your most up-to-date clicker scores this afternoon. So that's my current plan as far as that's concerned. But before that, we should actually get some new clicker scores, right? Everyone can add 2 more points to their clicker scores with any luck? So let's get started. The protein complex at a eukaryotic transcriptional pre-initiation complex that is nearly as large as the RNA polymerase, if not larger, that does not bind DNA is called TF2D, TF2B, TBP, the mediator, TF2H. I'm the stupid one. 15. Yeah, exactly. Well, it says bring the median score. 10. 5. Okay, polling stopped. So, couple of things on here. How can you eliminate the first 3 really quickly? Binding to DNA. So this complex is not binding to DNA. So that brings you down to D and E. Did we talk about TF2H binding to DNA? Specifically? Not really. I don't remember us talking about it. But it has helicase activity. And what does that mean? Got to be interacting with the DNA somehow in order to pull those 2 strands apart, right? So what does that leave us with? The mediator, which is fortunately what most of you thought, which is good. And we'll talk a bunch more about the mediator. Some of you may have seen the extra slide that I added in to the lecture today. We'll talk about that in just a second. But first we have to have another clicker question. Of course, have to. ATPase activity in the spliceosome is mostly required for blank activity, where blank could be DNA helicase, RNA helicase, endonuclease, exonuclease, or RNA ligase. 10. So here we are. We're done. And what do people think? Most of you thought RNA helicase, which is great because that is correct. It's the RNA helicase. We had some discussions in office hours about RNA helicases versus DNA helicases, and does RNA really form helices? Yes, most definitely does. And so people talk about stem loop structures. It should really be helix loop structures because all of these are forming nice secondary structures in the RNA, and secondary structure in the RNA means two strands that are base paired with each other. They don't base pair with each other in a parallel fashion. They're wrapped around each other. So it's always going to be a helix. And so it's the RNA helicase activity, which is most important here for getting the rearrangements that you have to have in terms of splicing and getting splicing to take place. So now, as I promised, let's take a look at the mediator complex. I heard the comment down here in front. It's the big ASS complex. That is completely correct. And in fact, this is excuse me, a paper which I just noticed. It's now just available online in Nature. So I always complain about having the lectures be up to date. This one's actually more than up to date because it hasn't even been published yet. But it's available online at the Nature website. And you can access this, by the way, through any computer that's logged on to the PSU network. So you can actually take a look at this. But this is a really incredible structure that was just published by Patrick Kramer's group out of Munich, where they've looked at the pre-initiation complex, which is this gray thing in here. That's your RNA polymerase, plus a number of these extra subunits, TF2B, TBP. It's missing the rest of TF2D because this is all stuff they put together in a test tube. It's got TF2F, doesn't have TF2H, but most importantly, it has about 2/3 of the mediator complex that's attached to it. So yes, this is big A in terms of the rest of everything else which is there. Everything which is blue on here, blue and violet, is the mediator complex. So not bound to DNA. You can see the DNA over here. This is the stuff that they had in their complex. These two pieces, everything's dotted here is what they think is extra stuff. So extra stuff here, extra stuff down here, extra stuff here. It's all stuff they didn't include in their structure. But it's humongous, this whole process. And this is just getting transcription started in eukaryotic systems. And so, again, hopefully, this is now really obvious that you've got a whole bunch of proteins that are here. And the DNA is really relatively minimal connected to the rest of it. And again, we still have a lot more to do because we don't know what the structure is of TF2H, TF2E, and the rest of the mediator complex. So these are the next steps that need to be done in terms of trying to understand what's going on here. Yeah?
The structure of the mediator, it looks like with like spine and arm, like maybe there's some conformational changes happening there when it binds with the other proteins at the spliceosome. So is there stuff like, does it have some kind of like ligand conformational change property, or?
So the question is, again, I'm sorry, I'm going to paraphrase here, but it looks as if, since you've got fixed jaw and movable jaw, maybe is one of the giveaways there, that this is actually some conformational changes which are going on in terms of what actually happens in the process. Is just a snapshot of that. And so to some extent, the answer is yes, definitely. There's stuff which is moving around. How we being this group here actually did this, got these data movable and fixed is lots of other data above and beyond this particular image, which is from electron microscopy. And this electron microscopy is taking, remember correctly, in this case, it's about 4,000 pictures of this complex and averaging them all together. But if you look at other pieces of data, you can see that those two pieces up here, the ones that are labeled as fixed and movable jaw, In some conditions, those are going to be right next to each other. Other conditions, they're further away relative to each other. And so those are clear conformational changes that we know are happening. And clearly, this whole situation is very dynamic. And so this is a great snapshot of part of the pieces in one particular time under one particular situation. And so it's clearly a very, very narrowly focused view of what's going on there. Fortunately, it allows us then to make hypotheses in terms of making experiments to try and understand what's going on in other conditions. But yes, you're completely right. This is only one snapshot of a very dynamic situation which is going on there. Okay, so let's shift gears a little bit. That was the pre-initiation complex. That was transcription. That's good review for the stuff you're studying for the midterm. What we haven't talked about more so far is looking at splicing. And this is getting back to looking at the spliceosome. And if you think about the consensus, quote-unquote, sequence for splicing, it's pretty non-consensus-like. There are lots of different sequences. The number of nucleotides that you find that are going to specify exactly where your splice is pretty few and far between. So how does the cell know, to over-anthropomorphize, what introns to use, or probably much more importantly, what exons to actually use? How do you do splicing? And how do you know what pieces of RNA you're going to want to put together? And that basically comes down to a couple of things. The first one is we really can't forget that splicing happens while the RNA is being made by the RNA polymerase. So it's always going to be going from the 5' end to the 3' end. So the most important things are going to be where you start out with. You know, what's your 5' start, and then where you go to a little bit 3' of that. So that's the first main thing, is that splicing happens while you're transcribing. You also get capping, which happens immediately on transcription. Yeah? So the question is this all happening by the C-terminal domain? And I'll paraphrase that and say right next to the C-terminal domain, because that's your tool belt. That's where you've got all of these splicing and capping enzymes that are associated with the polymerase. So yes, it's happening all next to the C-terminal domain, which if you go back here, they just have a little, this is where we think the C-terminal domain is in this picture. But that's more than they actually have in their structure. Sorry. So yeah, all of this is happening again. And if you thought that particular image was complicated, think about with the extra RNA coming out of it. There was no RNA in that structure. There's no C-terminal domain in that structure, et cetera. So these are really humongous, very complicated machines, really, nanomolecular machines. So you're making this RNA, you put a cap on it, and then you start thinking about splicing. Again, totally over-anthropomorphizing here. But the main thing seems to be not so much the introns, it's really exons. And so it's all about the exons as far as splicing is concerned. And that makes sense, because that's what you're going to end up with when you finally mature RNA that's going to end up being translated. So how do you recognize what exons are? Well, we'll see in just a second. Exons are pretty well conserved. They're usually just about the same size, 150-odd nucleotides. And, as we talked about already, these are where you end up with U2 associated with, so the beginning of the next exon, which also depends on that branch binding protein and the U2 activating factor, because those are getting U2 there. So U1 seems to be pretty normal. Okay, that sits down at the end of the first exon. But it's putting that U2 on, which is really, really critical. So we've got our U2 activating factor, which is important here. And then these purple blobs. Now, these purple blobs are also known as SR proteins. And this gets back to all those wonderful things you can remember from our discussion of amino acids. S stands for serine, R stands for arginine. And these are just the particular amino acids that you find a whole bunch of in these proteins. Arginines have what kind of charge, neutral pH? Positive charge. What kind of charge does RNA have? Neutral pH? Negative charge. Why? Because of phosphates. Because again, it's a phosphate ester backbone, just like you have on DNA. So probably those are involved with that. But they do bind very specifically to exons and not to introns. Introns have these wonderful RNPs associated with them. What's an RNP? A ribonucleoprotein complex. So RNA, protein, all together. And even better, HN, H is heterogeneous. So whole god-awful mixture of different things. So they're not well defined. And then what's the N stand for? We already had the SN RNPs. That N stood for nuclear. Right, so these are heterogeneous nuclear RNP complexes. Okay, so they're just things that are associated with RNA that are not nicely defined. They're not like SR proteins that you find lots of that are the same. They're not like our U1 and U2. Very nicely defined, always the same thing. These are just a whole bunch of different RNA binding proteins that bind mostly to introns, but occasionally to exons as well. So again, usually you don't want to have too many of these hnRNPs associated with your final RNA after splicing. On the other hand, you want to have all these SR proteins. So what happens if you have problems with this? And it turns out a lot of genetic diseases are due to changes in splicing. So one of my ex-students actually had an interesting genetic disease having to do with collagen called Ehlers-Danlos syndrome, almost definitely due to a spontaneous mutation that had happened that caused a splice site change in collagen. So what happens here, if you have splice site changes, most of the time you just skip an exon. You skip an exon completely because, again, usually there's some kind of definition of that exon by these SR proteins, et cetera. We'll take a look at that a little bit more later on, how that actually works. But most of the time, you don't recognize this exon, so you just completely skip it and go to the next one. Occasionally, you'll have these cryptic splicing signals. And again, that's because the consensus is pretty crap for splicing. And so you can end up splicing into a different place. And in this particular place, now there's another place that you can splice in exon number 2. Why does this work? Well, it mostly works because introns are pretty much always the same size. So look just between favorite model organisms, worms, flies, and humans. They're all centered at about 150 base pairs in length. Yeah?
Most introns or exons are the same size?
Oh, so exons here. Did I misspeak? That would not surprise me. So exons are the same size. This is why I put this up here to remind myself. Yeah?
In the previous slide, what happens when you have that cryptic exon spliced?
So the question is, what happens if you have a krypton exon splice like this? You end up changing your protein.
So it goes forward, and it's just not.
It can go forward. We'll talk about some of the quality control mechanisms a little bit later on. Okay, so exons are about the same size. Centered around about 150 nucleotides. 150 nucleotides gives you how many amino acids? 50? That's fine. That's why I have the mic and you don't. So it's about 50 amino acids. Just by chance, how big are most domains of proteins? 50-odd amino acids, 50 to 100 amino acids. So each exon very often is encoding for a single domain. What's a single domain do?
Specific function.
Has a specific function. Why? Because it has a specific structure. So these exons very often correspond to individual domains of different proteins. And so this alternative splicing or exon skipping can often change the domain structure of your protein, which is going to do what? change the structure, which changes the function. Exactly. So by the end of the term, you'll have that down. It'll be automatic. You're pretty close already. Good. So just by contrast, down here at the bottom, I wanted to remind you what I've probably said far too many times already, and that is exons are really nicely conserved in terms of their lengths, whereas introns are not. They are very widely spread between our different model organisms. Turns out human introns are generally longer than the ones you find in worms and flies, but a bunch of them are 5,000 to 30,000, and some of these are greater than 30,000 nucleotides in length. So keeping track of those is going to be a lot harder, whereas your 150-odd nucleotides is going to be really close to a nice number for your exons. What else is about 150 nucleotides in length? The nucleosome, what's getting packaged in the nucleosome. So it turns out that often, one or two nucleosomes end up being an exon in many, many cases. And that is going to become important just as we talk a little bit more about the interactions, of course, between transcription and splicing, even though might not be as obvious at the time. So as if it wasn't complicated enough to have just this one kind of splicing, it turns out that there are a number of different spliceosomes which are used in some cases. Not very common, but you certainly have some of them. This makes, again, predicting splicing that much harder because you have different sequences which are used. And so let's look here at the U12 type. U12, which is going to be another one of these small nuclear RNP complexes. It has an RNA which binds to conserved AU at the beginning of your exon as opposed to what U1 binds to, which is GU. The U12 actually binds to a slightly different sequence at the splice junction where this lariat forms, but it's still an A residue, and you still get this regular lariat formation. But again, the take-home message here, I don't expect you to remember the 1, 1, 1, 2, et cetera, is that we can have different sequences at the ends of your intron, which is yet another reason why what's in the exons is really extremely important. The last really pretty amazing example, which is only found out in about the last 10 years, is you can actually take two completely separate pieces of RNA that are not connected to each other on a single transcript and hook them together. And that works through, and it turns out, an extremely similar process for hooking these two different RNAs together, generating, again, a whole lot of diversity in what you can end up with in your final messenger RNA. These two, however, are much rarer than this process right here. So through this alternative splicing process, you can end up with, again, with these cryptic splice sites that we talked about before, but just normally in transcription, followed by RNA processing, ending up going to protein, people guess that about 60% of the genes, starting out something like this with exons and introns in it. The introns here are actually probably far too small, relatively speaking. I wonder if this is actually mislabeled. I would think the exons would be these individual ones here. Go back and take a look at that. But here, the exons and introns, depending on which of these exons you end up using, you can end up with very different proteins in different, very often, cell types. So different muscles, striated muscle, smooth muscle, fibroblasts, brain. You have different splicing that's all happened from the same gene here, giving you different domain compositions and, of course, different domains, different structures, different functions. A couple other things to notice here. One is that most of the exons are the same for all these guys. It's just a couple of them that are different. So here at this end, this final 3' exon is different There's a couple of different exons that are used here. This one is used in one case, but most of the other ones here are exactly the same. All of these exons are identical to each other. You have a couple of minor differences which are here. And about 60% of genes in humans, that's guess, are alternatively spliced, and about 80% of the time you could predict them. That means 20% of the time we have no clue. And that's 20% of the time of these 60% which are alternatively spliced. Alternative splicing is really pretty amazing in that it can generate an absolutely ridiculous number of possible genes, possible mRNAs from one gene. So this is an example in Drosophila. We actually know that more than 38,000 different splice products can be generated from one gene. So we may only have between 20 and 30,000 genes in the human genome, but if each of those could be producing 38,000, although this is a really extreme case, you could have 30,000 times 30,000 genes in terms of the actual different proteins that you end up with. So it's a really huge number. But again, just like in this case over here, the human case for the tropomyosin gene, in this Drosophila gene, it's also mostly the same exons that get used, only a couple of different alternatives. And the way that you get to this 38,016 is you just multiply 12 alternatives by 48 alternatives by 33 alternatives by 2 alternatives. And that way you can end up with all of these different possibilities. Few alternative exons, which can then lead to very different functions at the end. So mentioned exons are usually about the length of one nucleosome, and splicing is co-transcriptional. You've got the mRNA coming out of your elongating RNA polymerase complex next to the tool belt, the CTD, which has all these proteins on it. It turns out that it's not terribly surprising that the splicing, and particularly alternative splicing, which of those exons you use or don't, depends on how fast the polymerase is moving along the DNA. And how fast that polymerase is moving along the DNA is going to depend on your chromatin. So if you've got heterochromatin, you've got things that are compacted, it's going to go slower. And if it goes slower, those splicing enzymes actually have more opportunities to be interacting with that messenger RNA that's being made. So if you're going slower, you're more likely to use more of those alternative splice sites that you have there, and you end up with more splicing than you do if the polymerase is cruising along the DNA, making RNA at a really fast rate. Because that RNA skims by before the splicing enzymes have a chance to jump off the tail and jump onto the RNA, you may have missed some of those splice sites. And so that is one way that you have a change. Another interesting thing that people have found, we don't quite understand why this is the case, but there are a bunch of modified histones that get added to the DNA. Remember, so this is splicing happening on the RNA, but these are changes that are happening on the DNA relative to where you have splicing and where you don't have splicing. What those exact interactions are is not entirely clear. There may even be some of those spliceosome proteins that are associated with some of these modified histones. And this is getting back to when we talked about the histone code before. It's not just euchromatin, heterochromatin. It's also involved in interactions with other proteins. Yeah?
So you said, just really quickly, so if it's moving slower, it has more time to interact with the RNA?
Yeah, so if the polymerase is moving slower, the RNA moving past the C-terminal domain is going to be going a little bit slower. And it's not as if it's going slower versus faster. The way, and I mentioned this before, but I probably should emphasize it again, is that the RNA polymerase just doesn't go at a nice constant rate, faster or slower, but it sort of goes and stops and goes and stops and goes and stops. And we know that, again, from looking at actual elongating complexes. And so if you're sitting around stopping for a while, then maybe the splicing complex will be jumping off the tail and getting onto the RNA, which is right there. Whereas if you're moving along and not stopping very long, it doesn't have a chance to jump on and do its thing. Yeah. So the question is, when do those histone modifications actually take place? Are they present there before the polymerase is chugging along and does its thing, or are they present after it's done that? And the answer is yes. So both. There seems to be some before and some after. And it's particularly those after which are really weird, and we're not quite sure why they're there. It sort of said, hey, we did alternative splicing here. Okay, what's the point? And so maybe it's if it's a gene which has just gotten turned on at one particular point in time, then you have alternative splicing that happens there once, but then that says you're in a particular cell type and you want to be sure that that alternative splicing happens much more regularly, so that could be a signal for doing that. Those are really hard experiments to do, and so that's one of the theories, but no one's really tied that down yet. Yeah? So if anything, again, it depends on how you count. Sorry. So to clarify, the faster you go, the more alternative splicing you have. I wouldn't say that per se. I would say the faster the polymerase goes, the less likely you are to use each of the particular individual splice sites. It's a slight paraphrase there. Yeah, so let's look at, that's, of course, one way that these things are happening. This is just, I wanted to get another image other than the ones that are in your textbook, because I think it doesn't do a terribly good job of looking at this. This is from a review article. Sorry I don't have the link here to this particular one. But this is looking at, unfortunately now they call them RS proteins rather than SR proteins, and how they're interacting with exons. So here's the cartoon of the U1 snRNP bound at the 3' end of the first exon, and then U2, which is going to be binding at the branch point towards the 3' end of the intron. How it gets there, it's these interactions with U2AF and really these RS or SR proteins, and again, that just stands for arginine and serine, that are common amino acid sequences in these proteins. If they're bound to the exon, you're going to use this exon because you've put U2AF and got U2 binding. On the other hand, if you don't have these SR proteins bound here, often because there's a different protein which is bound there, then you don't use this particular exon. And these are also what are known as splicing enhancers. It's what this purple sequence is supposed to be here. Purple sequence, if it's bound to specific protein which blocks splicing, blocks the association of the SR proteins, you're not going to get U2AF, you're not going to get splicing. In the absence of this protein, your SR proteins bind here, you get U2AF, you end up getting splicing. Because of this, you can get lots of different alternative splicing that can happen, and this is getting back to your question, faster alternative splicing, not alternative splicing. It's more about how frequently you use any of those given splice sites in terms of how fast. And it's not so much how fast in general the polymerase is going, it's how fast the polymerase is going at the point that that particular splice site is being transcribed. So you can get what they call skipping exons, what we talked about already. You can have different sites at the 5' end that you get. And the 5' end here, we're talking about the intron. So 5' splice, you can have alternative 3' sites, and all of these are changing what you end up with in your final messenger RNA, which you have over here. Sometimes you even hang on to introns. So it's a piece that gets spliced out that actually is coding sequence that sometimes is used, sometimes isn't. Very often you'll end up with this either one exon or another exon. If you go back and look at those alternative splicing examples that we had before, you'll see that it's not terribly often that you end up with one extra exon. It'll be one exon or another exon which is there, or again, this skipping of exons which takes place. And then there are even some examples down here at the bottom, sort of the most extreme one, where you can end up with a much shorter messenger RNA, depending on your splicing, rather than a much longer one. And we'll see why that is when we talk about poly-A tailing in just a couple of minutes. This is not just an academic exercise talking about this. There are many cases of human diseases, as I mentioned before, where you have changes in splicing. And this is because you can have one or very few nucleotide changes, which can cause a huge number of changes in your final messenger RNA. So you can be introducing stop codons, you can be getting frame shifts, you can get a totally different protein which is being made just because of one little nucleotide change which is changing splicing. And this is quite well known in a lot of blood diseases. One example here, and these are from real patients where they've looked at the messenger RNAs. Most of the times these are in, heterodiploids, and so you've got one copy that has a aberrant splice and the other copy is normal. In most cases where you have a homozygote, then that's lethal, and so you're never going to see these particular kinds of patients. But in heterozygotes, you'll often see these changes, which cause lots of different, and these are relatively easy to see. You just pull out white blood cells and take a look at their RNAs, how they're being made. So some cases, exons are skipped, and this is particularly in the beta globin gene. Few cases, you'll have a single nucleotide change, that gives you a different 3' end to your intron, and in one case it actually adds an extra exon in your sequence. Turns out that, not surprisingly, this particular gene leads to a much more diseased state than this one here. And if anything, you could think about maybe this was an original state and then you got a reversion of not that one particular nucleotide change, but another one which brought it back to almost normal. So you just end up with a little bit of extra amino acids which are there. So these, again, are diseases that we know of, and these are specific examples. Yeah?
So in the case of C,
yeah.
that would be a mistake or an error in transcription that would lead to that single nucleotide change?
So, okay, the question here is what leads to this single nucleotide change? It could be a single nucleotide change that happens because transcription is taking place and the RNA polymerase is not very high fidelity, and we'll just missplice that one. So it gives you a one messenger RNA which is incorrect. It's highly unlikely that we'd actually see that, because that's going to be one messenger RNA which gets differently spliced and be one protein. We'll never see that. So these are almost always genetic changes which have happened due to some kind of DNA damage or some kind of replication error. But it's at a DNA level that you usually see these different changes. Yeah?
Could it ever happen on purpose as a result of environmental change, like in one case, one kind of splicing is good and then something else changes?
Okay, so I guess the question in here, again, I'll paraphrase, but that are there environmental effects on splicing? And so you have one particular kind of splicing under one condition and a different kind of splicing that happens under another condition. And the answer there is definitely yes. Exactly what those conditions are and how those are being sensed by the cell is a really open and interesting question. We'll talk about some regulated splicing later on when we talk about gene regulation. So it's the fact that you have all of this splicing taking place means it's an opportunity for regulation. And so it allows you to have a lot more diversity in that way. So one of the big questions that many people ask is, why is it all RNA? Well, it's all RNA because RNA is probably what was first around in terms of actually doing all these things in the first place. And when we talk about translation, it's all about the RNA. The ribosome is almost completely RNA. Proteins are probably a relatively late arrival in the whole process of what makes life. And then Dr. Lehman in the chemistry department, this is the whole focus of his research. People also talk about the RNA world, and we'll talk about that in Wednesday's lecture, maybe in Friday's lecture a little bit more. But basically what the idea is of the RNA world, and we know quite well that RNA can actually be enzymatic in terms of enzymatic activity. So pretty much all enzymatic activity can be done by an RNA. It's usually not as good as proteins, which is probably evolutionarily why we have a bunch of proteins right now. But RNA is really all you absolutely need, probably for some pretty basic life functions. So not surprisingly, you have to generate diversity in that RNA. Well, what's one way to generate diversity in RNA? Is splicing. And it turns out that there are quite a few what are so-called self-splicing introns. And a self-splicing intron is a stretch of RNA that that RNA by itself can chop the RNA out and fuse what's on the outside to the ends. And a number of people, particularly Dr. Lehman, again here in the chemistry department at PSU, think this is how replication originally started, making RNAs. And so a self-splicing RNA can basically make more of itself by hooking ends together. We talked about trans-splicing before, pretty rare in cells now, but could certainly have been happening probably billions of years ago. Turns out that there are two different kinds of self-splicing introns that we know of now. A few exceptions to that, but the two major ones, they have very different structures, which is why one's shown as a hand, the other one's shown as a foot. But again, they have these 2' OH structures here that form lariat structures, or in one case there's actually a G which gets added to the RNA, and that G ends up being the place where you form this lariat-like structure. So self-splicing introns, again, it's an RNA. You can make that RNA. You can make it in a test tube. You purify that RNA. You put it under the right conditions, and lo and behold, poof, chops a little piece out, puts it back together. This is a really fascinating reaction, how it actually takes place at a chemical point of view, but it's kind of a fun story that goes with this. The guy who discovered these things in the first place, or I should say the head of the lab where they discovered this in the first place, his students kept bringing him these results and said, hey, I got rid of all of the protein in here, but these things are still splicing. He said, no, go back and do it again. I don't believe you. And go back and do it again, go back and do it again, go back and do it again. Eventually got the Nobel Prize because it really was just the RNA which was doing this. And that was the original one, was in fact these group 1 self-splicing introns. Turns out there, again, there's a whole nother group which turns out the mechanism here is almost identical. So we talked about RNPs before in terms of getting splicing. So the ribonucleoprotein complexes, you don't need that P at all. You can just do it with the RNA, which gives you this splicing. Much more efficient with all those proteins, many of which are what? Hydrolyzing ATP and serving as, that was that clicker question, like, you know, helicases, yes. So many of them serving as helicases. Turns out that some RNAs can serve as helicases as well, but proteins are way better at forming helicases. And that's probably why the current spliceosome process is much faster and much more efficient. It's because you have those protein helicases, which are there. So we'll talk more about RNA world stuff again a little bit later on. I think it's really fun and great to speculate on what was going on 4 billion years ago, maybe on Earth, maybe on other places. So we'll get back to that. But first, let's talk about what happens to the 3' end of your RNA. 3' ends of RNA, what happens to them? Eukaryotes, you get a poly A tail. How does that happen? Well, it turns out, again, it's all to do with what's present in the RNA sequence as it's being made. There's a quite well conserved sequence here that happens 10 to 30 nucleotides upstream of, again, a pretty well-conserved sequence here. Then a much less well-conserved sequence, which is present over here at the 5' end. What happens is you get cleavage. So this is an endonuclease that happens in the RNA as it's being made. Give you one guess where those proteins are. Let's do this. They're hanging on to the C-terminal domain of the RNA polymerase, exactly. So as this messenger RNA is coming by, they bind to these particular sequences and lead to endonuclease. Once you have this endonuclease cleavage that takes place, you put on a whole bunch of A residues. This is the poly A tail, usually around 200, varies from species to species. And this is now your final messenger RNA that gets pumped out of the nucleus and leads to being translated. Yeah?
So with the use of that tail, that enables or does that enable the process of transcription to be continuous, so it doesn't have to back up in its directionality? It can just keep going, and then the tail modifies as it moves through? That new mRNA is being synthesized?
Okay, so I'm going to try and paraphrase your question here. But so the idea here is that the RNA polymerase could potentially keep going. So this would be one messenger RNA that then gets released, and then this will be your next messenger RNA, and you continue and you keep going.
Yeah, I'm just talking about directionality.
Right, and so again, you're making, this is here, your RNA polymerase is over here on the right-hand side of the slide. It's continuing to move along here. It's continuing to transcribe. And so then you have this cleavage event that takes place here. What's actually different, because we talked about polycistronic and monocistronic RNAs, what do you usually have in eukaryotes? Monocistronic RNAs. So it turns out after you have this cleavage event that takes place, there are a bunch of hungry exonucleases which chew this up. And so they end up, so your polymerase is going along making the RNA. It gets chopped off. You make the poly A tail. But then there are exonucleases that basically chase down the polymerase and chew up the RNA and chew it up in such a way that now that RNA polymerase falls off the DNA. And so we talked last time about termination. Actually, not last time, a couple times ago, about termination. In bacteria, you have the speed bump in the oil, your helix with a loop at the end with the U tail that sort of pops itself out of the RNA polymerase. Here, for eukaryotic transcription, it's very different. What seems to happen is it's that exonuclease activity, which is chewing in at the end of this newly released 5' end. It's that degradation which seems to lead to dissociation of the polymerase and termination of transcription. It's a very different mechanism for how you're getting termination. And it's just one that you get now. It's just that monocistronic RNA. Again, mentioned there are some polycistronic messenger RNAs in eukaryotes. Again, this is the major case here. Exceptions. So let's look at what does that. Again, these are our friends sitting here on the C-terminal domain of the RNA polymerase as it's transcribing. Here's that messenger RNA coming out of the polymerase. Once you have the transcription of this RNA, that first protein, called CPSF, so it's got an F at the end. What does that mean? Factor, probably a protein. It's the cleavage and polyadenylation specificity factor, CPSF. It actually first binds to TF2D, gets associated with the pre-initiation complex, and then jumps onto the C-terminal domain. That's the first guy that interacts. That's actually with this sequence, which is now 5' to where your cleavage site is, and then the cleavage stimulation factor, CSTF, is going to bind to the sequences which are 3' to where you're going to be cutted, and this stimulates cleavage. And I'm not going to ask you what the names are of all of the endonucleases. I'm just going to expect you to remember the things here which are nicely in red, conveniently for you to remember them. And then this stimulates that cleavage event that we just showed you here. You have the poly A polymerase, which jumps onto the 3' end, puts a whole bunch of A's on the end, And then you have, again, the incredibly creatively named poly A binding protein, which binds to this poly A tail. And this is now the 3' end of your RNA. After this cleavage event, as I mentioned before, there's now the exonuclease, which jumps on here and chews everything up at the end. So it's all about this C-terminal domain of the RNA polymerase. It's not just CTSF and C cleavage and polyadenylation specificity factor, CPSF, CSTS, CPSF, alphabet soup. Really useful thing. Write these down on a big piece of paper and write out what all these abbreviations stand for. So it's not just these 3' end processing proteins or the capping proteins, the guanylyl transferase and the methylase enzymes. Also have all of your splicing proteins that are associated here. And again, not surprisingly, it depends on how fast your RNA polymerase is moving along the DNA, which is going to depend on the nucleosomes which are there. All of this is that CTD, which is hanging out there. So again, it's the tool belt which is there. It gets phosphorylated by TF2H. That process is also involved in escaping the promoter. So here's your phosphorylation of the CTD. That phosphorylation also seems to be important for getting these other proteins bound. So all of the splicing, capping, tailing enzymes don't bind to just the CTD by itself. They're only going to bind there once you have phosphorylation that takes place. So again, a central role for TF2H in this whole process. And a lot of people talk about this as the messenger RNA factory. So it's not just making the messenger RNA in the first place. It's also capping, splicing, and tailing it, all of which is in this elongating polymerase. And some of the examples here are just looking at, here's our spliceosome, here's our capping enzyme, all of these associated with the CTD. And I like this slide because it gives you an idea of how long this tail is relative to the size of the polymerase itself. So it's pretty ginormous. Is that a stretch or a question over there? A stretch. Getting towards 10 o'clock and it's Monday morning. My daughter really didn't want to go to school this morning, so I understand I'm in the same boat. So yet another way of looking at this, we've got the tail of the RNA polymerase, all of these proteins that are involved in transcription, messenger RNA processing. But if you look at that complex, so you think, humongous RNA polymerase, big mediator complex, now all of these extra proteins that are associated with the C-terminal domain, that's actually way bigger than the poor little DNA, which is at the bottom of it. So it actually makes much more sense from a topological and geometrical point of view to, instead of having this whole complex moving along the RNA, see here's my RNA or DNA here, a nice little DNA complex, it makes much more sense, since this complex is ginormous, to actually move the DNA through it. And in fact, that's exactly what you see if you look in nuclei of cells that you've stained with some of these complexes. You see a bunch of spots. If all of these guys were moving along the DNA, which is spread all the way throughout the nucleus, then these would just be big smears. But what you see are very defined spots inside the nucleus. And what that tells you is that all of these guys are actually attached together. So you'd have an RNA polymerase here with its tail that's associated with a bunch of other polymerases. Here's another RNA polymerase with a tail. Here's another RNA polymerase with a tail. All of them brought together in one part of the nucleus, and you're pushing or pulling the DNA through that. It's not that that whole complex is zooming along the DNA. And again, it makes sense because there's a whole bunch of different proteins which are there. And it's a lot easier to move the small piece, which is the DNA, than it is to be moving this massive complex. So all of this is present together in the factory, which is not just one messenger RNA assembly line. It's multiple different messenger RNA assembly lines that are all together in one place. So you've got your Silverado coming out that way and your Charger going out that way, but it's all using a lot of the same processes which are happening. Grew up in Michigan, so I have to use that analogy. So last thing that I wanted to talk about in terms of messenger RNA, we've made it. It's got a cap, it's got a tail, got all the good splicing that's happening, but you can't translate it yet. You've got to get it outside the nucleus before you can actually have translation take place. How does that happen? What's mostly due to the association of all of the appropriate proteins that are on your messenger RNA. And we talked about quality control a little bit earlier on. This seems to be one of the most important quality control steps, is you want to make sure that all the splicing has taken place and the tailing and the capping before that RNA gets sent out into the cytoplasm to get translated. And as we'll see at this rate next time, that's an incredibly energy-dependent process. Translation, as it turns out, is one of the most energy-demanding processes that the cell does. So you just want to translate the right things. So you want to make sure that any of the messenger RNAs that are incorrect don't get exported out into the cytoplasm. So there are a couple of ways of checking that. First one is the cap binding complex. So that makes sure that the cap is formed properly. Your 5' methyl G, which has gotten hooked up by that 5' bond to the first nucleotide of your transcript. You lose almost all of these heterogeneous nuclear ribonucleoprotein complexes. Where are those mostly bound? Introns. Do you want introns in the RNA you send out? So this is the way that you're checking to make sure that you've gotten splicing that's taken place. One of the things that I didn't mention before, but after splicing, you end up with these boxes. They're not really, of course, a box, but the exon junction complexes. So it turns out when you put two exons together, there's that splicing spliceosome process then brings the exon junction complex to these two exons that have been put together. And this is a really good indication that splicing is taking place properly. So having exon junction complexes is also extremely important. We already talked about poly-A tails and poly-A binding proteins. So you've got all your exons put together. You've gotten rid of your introns. Again, the exons are together properly, which you know through these exon junction complexes. That then binds to a nuclear export receptor, which people in cell biology talk a lot more about. And this nuclear export receptor then pulls everything through the nuclear pore complex out into the cytoplasm. In the cytoplasm, this cap binding complex comes off, and then you start translation. Once you've done this, of course, you've had all the splicing taken place, you've chopped up your RNAs in the nucleus. You end up with a whole bunch of RNAs in the nucleus that you don't really need anymore. These are the extra bits, the introns, sometimes thousands of nucleotides long. So you put these into the nucleic acid garbage disposal. That's this exosome complex. And basically, it's a whole bunch of exo and to some extent endonucleases, but mostly exonucleases, which will chew up any extra RNAs down to the individual nucleotides. Those then will get recycled, and everything stays in the nucleus, and we're all happy. Yeah?
When we talk about the sort of quality control checks before the mRNA leaves and the loss of HNRNPs associated with that, what about when the mRNA, like, it's specific, like that example we talked about where introns were specifically included in the mRNA?
Yeah, so the question is, how does quality control work? Again, I'll paraphrase. How's quality control working when you have like an intron included example? We talked about the alternative splicing. That's a very rare case that you have these introns included. And in that case, there actually aren't any HNRNPs that are bound to that particular intron. And so that's generally not a problem there. We'll talk more about quality control as well. It turns out there are a few cases where you get RNA export that takes place, where you have incorrect splicing, you don't have proper tailing. We'll talk about how you can control for that when we talk about regulation a bit later on. Okay, so in the last 5 minutes, I wanted to talk about some of the RNAs which stay in the nucleus, because RNAs are really important for translation as well. But how do you get these RNAs that then get exported and then used as functional RNAs out in the cytoplasm? Well, it turns out that ribosomal RNA, how much of the cell is it? Approximately 80%. So you have a whole bunch of it, which is there. How do you make it? You make it with RNA polymerase 1, not RNA polymerase 2. And you have many, 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 many copies of these genes. That's how you can get so many, how you can get 80%, which is actually there. This particular ribosomal RNA gene is also made as a precursor, just like all the messenger RNAs are made. But turns out that this gets chopped out as opposed to being spliced. And then, much more importantly, it gets highly modified. All of these triangles and circles here represent modifications, but it's a really small number of the modifications that actually take place. It's just supposed to be being shown there. And what does this? It's the RNAs. So we had our small nuclear RNAs that were important for splicing. Now we've got our small nucleolar RNAs, which are important for this cleavage and modification process. The first one, everybody remember U1, U2, U4, U5, U6? We skipped one, right? That's U3. Well, U3, it turns out, is important for ribosomal RNA processing, and it makes the first cut that happens at the 5' end. So now we've done U1 through U6. Now we're happy. But now we have all these small nucleolar RNAs. What are small nucleolar RNAs doing? Turns out the small nucleolar RNAs are what you need to get the individual modifications that you have to have in your ribosomal RNA for it to work. How do these guys work? There's multiple different ones, but there are two main kinds of classes. These are called the box CD and the box ACA SNO RNAs, and that's just because they have either specific sequences, ACA and these box HACAs, and then CD, just the sequences which are well conserved. And so if you think about conservation, we already talked about -10 and -35 and eukaryotic promoters. People also talk about these as boxes as being well conserved. Remember the Tata box? That's because they conserve sequence. So these boxes are conserved sequences. The CD box SNO RNAs lead to O-methylation of ribose. So the 2 position of ribose gets a methyl group that's added, and the HACAs gives you pseudouridine, which is basically uracil, which you flipped over, and has given you, instead of the NH, which is normally bound over here, now you have the C and CH, which are bound over here. Basically just flipping that over. How do you know where to get these modifications to take place? It's because all of these SNO RNAs have specific nucleotides in them that direct the modification to one specific nucleotide in the ribosomal RNA. So it's through base pairing interactions to get the SNO RNA binds to a specific position in the ribosomal RNA, leading to that one modification that takes place. Other modifications happen as well, but the main ones are 2' O-methyl ribose and pseudouridines that take place. So that's what happens in the nucleolus. And now we've done messenger RNA processing, and it's 10:05.